Dear audience, my name is Roland Fejes and let me speak a little bit about sepsis and about our research work in the Institute of Surgical Research. The date you see here indicates a historic moment because on this day a medical doctor and a physicist conquered the highest peak of the world, the Mount Everest. It was a tough project because today every alpinist knows that above 8,000 meters there is a death zone where the oxygen saturation of the blood decreases rapidly and in 3 minutes death occurs. But what was the discovery that made this result possible? One of the most obvious things in the world, it's the oxygen. Oxygen is essential for our survival. Just like digestion has a dedicated organ system, so does the air exchange. Through our airways, oxygen reaches the alveoli where respiration takes place, and then through macro and microcirculation, oxygen reaches the end user, the cells, including a cell organ called mitochondria. The mitochondria are also commonly called as the energy factory of the cell because they make energy from nutrients you may already know in the form of ATP. Based on this, our body has a certain oxygen supply and oxygen demand, which is in good case in balance with each other. However, for example, in respiratory failure, the supply decreases or demand increases in inflammatory conditions. But what happens if these two phenomena happen at the same time? The human organism goes in the oxygen depth, a critical state, and this is exactly what happens in sepsis. Sepsis, or as it's popularly called, blood poisoning, is based on an infection that invades the bloodstream. This triggers a dysregulated inflammation that disturbs the body's regulatory mechanisms and creates severe oxidative stress. This damages the microcirculation and the mitochondria, which is called microcirculatory mitochondrial dysfunction syndrome, MMDS, and the end state is a multi-organ failure. But what do we see in the practice? Infection, of course, but it can have many sources. In case of pneumonia, the patient coughs, has shortness of breath. In case of urinary tract infection, the urination is painful, and so on. The immune system works, so the patient will have a fever. Due to the released toxic substances, the blood vessels dilate, resulted in the decrease of the pressure of the blood on the vessel wall, and the blood pressure decreases. This is called arterial hypotension. The heart tries to maintain the blood pressure by increasing its frequency, but this leads to rapid exhaustion. I mentioned earlier that the poorly functioning microcirculation causes an insufficient supply, so nutrients and oxygen cannot reach the cells and the end products would not be removed. Ischemia and or hypoxia is developed, which directly causes damages on the mitochondria. In clinical practice, the so-called sequential organ failure assessment SOFA score is used to assess the severity of septic condition, which is a standard scoring system based on the estimation of the organ functions. How does this work in the practice? Imagine two patients. The first patient is healthy, who has almost physiological or just slightly worse results. If you add up the points, you can see what is the chance your patient will survive. Now let's take a septic patient with much worse organ functions. He scored higher and his probability to death is also much higher. It's an easy to use scoring system for humans, but the scoring system has not been adapted to experimental animals yet. The treatment of sepsis is basically symptomatic and it's called supportive treatment. The patient is ventilated with additional oxygen and the circulation is supported with fluid therapy. This is relatively useful, but often not sufficient as the ultimate victim of the process, the mitochondria, are not halved in any way. Thus, there has been a recent need to develop a mitochondria-targeted therapy. So the aim of our work was to establish A well-usable rat organ severity scoring system, the so-called ROFA score, after setting up a standard septic animal model on which we could try novel MMDS-targeted treatments, and we had 2 therapeutic targets to achieve this. For our experiments, male rats were used, which were assigned into control and sepsis groups. Animals in the control group were injected with saline, while a freshly prepared fecal suspension was injected into the abdominal cavity of the septic animals. Fluid therapy and analgesia were then given to the animals, and 22 hours after sepsis induction, the therapies were applied. On this slide, you can see the position of the animal. After anesthesia, it received fluid in its vein on the neck, and from there, we could take blood samples and give the pharmacons. Blood pressure was measured in the carotid artery, and blood sample was also taken to determine the amount of oxygen and carbon dioxide in the blood. At the end of the experiment, the abdomen was opened and video records were taken by using an intravitreal video microscope camera from the surface of the small intestine to determine the microcirculation. And finally, mitochondrial function was measured from liver tissue samples. Our first therapeutic target was the endothelin systems. Endothelins play an important role in the regulation of circulation as they induce vasoconstriction, inflammation, and increase permeability through the A receptor. The B receptor has 2 forms with opposite effects. We hypothesize that we can attenuate the symptoms of sepsis by inhibiting the effects of the A receptor and enhancing the vasodilation induced by the B1 receptor. And to achieve this, a combined endothelin receptor inhibition stimulation regime was used. Here you can see our results. We observed a very low blood pressure, hypotension in the septic animals, but the blood pressure increased significantly in the treated animals due to the vasoconstriction effect of the combined endothelin antagonist treatment. It means this deviation is large enough to know that it's not caused by just coincidence. The next figure shows that in the case of chemocurated animals, almost 100% of the capillaries were opened and perfused, while the perfused capillary rate decreased to above 60% in the septic animals. However, our treatment significantly improved the microperfusion in sepsis. A similar result was obtained also at the case of mitochondrial respiration. On this slide, you can see the results of the ROFA score. In sepsis, this value rose very high and it decreased spectacularly with treatment. It was not significant, but a half-hour-long treatment could not be expected to completely ameliorate the effects of 22 hours progression of sepsis. Our next therapeutic target was the kyrenic acid, which is a side product of the metabolism of an amino acid called tryptophan. Kyrenic acid acts on many receptors, but it has important cell and endothelium protective effect. The synthetic analog of kyrenic acid is the SR72 that was developed to enter the brain via blood-brain barrier, and it also has a very strong anti-inflammatory effect. If we look at the result, we can see that none of these pharmacons affected the blood pressure. In the case of microcirculation, SR72 was not effective, but kyrenic acid was able to significantly improve it. And in the case of the mitochondria, we see the exact opposite effect. And what happened to the ROFA score? As we see, SR72 had no effects. However, kyrenic acid was able to decrease the value of ROFA score, hereby to improve the organ dysfunction. And finally, what is the take-home message? The rat-specific organ failure assessment score is a very usable way to determine the severity of the septic multi-organ failure. As we have seen in the case of the endothelin regime, although we targeted the microcirculation, mitochondrial functions were also improved, which again underlines the fact that these functional systems are really closely connected. In the case of chirurenic acid and SR72 treatments, even though they have very similar molecular structure and we thought they have very similar mechanisms of action, the results are completely different. Chirurenic acid improved the microcirculation, while SR72 improved the mitochondrial functions. But however, all of these just highlights the beauty of science because hypothesis and reality often do not overlap. And this is the time when the rethinking of what may be behind this comes. Thank you for your attention.